That is why the Healing for the Ages Conference was established. So if you struggle with high blood pressure, diabetes, cardiovascular disease, if you struggle with neurological disease or symptoms, if you've been diagnosed in the past, are currently in remission, or have metastatic diagnoses of cancer, that was why the Healing for the Ages Conference was established. So think about those people, yourself included, and anybody around the world who are struggling with any of those symptoms, and ask yourself, have they been given the solutions to those symptoms or those problems where they absolutely can live free of those symptoms without treatment? That's why the Healing of the Ages Conference was actually established. So this purpose of this next hour, and I really do appreciate all of your patience with allowing us the time to get this back online. We actually came here, tested this yesterday for an hour, and made sure this live stream feed worked, and it worked perfectly until the very minute we went to launch this tonight. And we were worried about that this whole time. So thanks for the team being able to pull that together now. Yeah. All right, so for the next hour, I'm going to personally start out, then Dr. Group, then Dr. Ely, then Dr. Schmidt. We're going to be discussing principles to set the stage for what you're going to be learning are the causes for illnesses and disease and symptoms, even the ones you're experiencing long-term since even having mild cases of COVID-19 4 years ago. And we're going to be presenting to you all the solutions along the way throughout the conference, Friday, tomorrow, and Saturday. But to set the stage and for the confidence you might need to decide to actually register for this event, if you haven't already, we're going to show a few slides here. I will start out for the first 20 minutes or so, and then we're going to pass it down to Group Ely and Schmidt on specific topics that may be of interest to you to help solve the riddle to why it is you or your loved ones struggle. And then is there a possibility that the Healing for the Ages Conference would be right for you to attend? If any of you actually decide after this actual Q&A tonight in Masterclass, if you decide to register, this is something very unique to this conference that I have never experienced at any other conference in my entire 20 years as a clinician. Neither is it for Dr. Group, Ely, or Schmidt. And we've attended conferences, spoken at conferences for over 20 years, each of us. Never have I attended a conference where the solutions were given, problems stated, root causes for those problems were actually explained. Never did I walk away from that conference and then receive in my email after the conference was over every single PowerPoint slide, presentation, research article, and link, and the solutions provided to you to take with you and have at home, so you didn't have to worry while attending the conference. Did I capture with my phone what they put up on a slide? Did I write it down fast enough and correct the doses and recommendations that they gave in that conference? No, you get to sit back and learn from individuals who absolutely have over 100 years of combined natural healing with patients in their own clinics for over 100 years combined. We are not intimidated. We do not have any kind of belief system that we are inadequate to bring you this information. We have watched the grand power and mastery that God put into our human body, which we consider to be the greatest of all of God's creations, that I believe he saved for day 6, which was his greatest of all. Prior to that day, he put everything on the earth for the benefit of man. That included the minerals in the dirt, the plants he established and seeds he put in that dirt, which provide the vegetables, fruits, food for all of us, and herbs. And then he put the animals on the planet for all of us also. Those resources were what he put down here and declared, I gave that to you for the benefit of man. After that is when pharmaceutical drug companies, drugs were manufactured, vaccines were manufactured, all synthetic. They put no trust and faith in nature. And that is why I assembled this team of naturopathic-centered individuals to help me once and for all provide a foundational conference to educate you what's going on in the world where science is both deceiving us and hurting us with their weapons. This truly is a healing for the ages of bioweapons. And that's what you're going to learn here. And there's more than one vector. So what I'm going to start out with here over the next 10 minutes or so, and I don't know if we're keeping track of the time. Do you remember what time it started? No, we need to know. Because I was asked to go to a conference one time and speak for 6 hours straight. So I don't want to get caught up in that. I want to make sure we stay on time for an hour. But since we were delayed, I think I'm at about 5 minutes. All right, so about 5 minutes in, so I got about 15 minutes here. There have been conversations, docs, that seem very controversial to some, including the medical profession. They don't really want to look at this topic. They have a hard time rationalizing how this is possible. Yet, if I asked anybody in this country any layperson, any medical professional, if I asked them, if you had a loved one who was raped and murdered, and you were suspicious to find out who did that to their loved one, you would put your faith in a courtroom and in the judicial system with an expert witness who would be an expert in which field? Genetics. And we would put our faith in what they call DNA testing. It wouldn't matter what the medical doctor found, what a chiropractor or podcaster declared might have been the cause of death. No, we would actually look for DNA evidence and then trust that evidence to perfectly confirm who it was responsible for the injuries and death to your loved one. Well, in the beginning of COVID-19, geneticists, DNA experts in China in January of 2020, they actually went to look at the blood work of the Wuhan sick patients, all of them, in hospitals in Wuhan, China, that they were identifying with a new COVID-like pneumonia. They took their blood, ran their antibodies, and determined, geneticists did, DNA experts, actually declared in January of 2020 that the origin of COVID was actually venom of two snakes, the king cobra and the Asian crate snake. From DNA testing, they were able to determine that the third most likely source came from bats, but it was never bats were the original. It was actually confirmed to be two snakes. 4 months later in France, in April of 2020, they isolated what were called, geneticists did, they isolated what was called the spike protein on COVID. And if you docs all remember, they said the only thing different about this new virus called COVID and this discussion of possibly that it was made in a lab with gain-of-function testing, that was suggested because snake venom had never been found on a previous coronavirus, these spike proteins. And they said this must have been manipulated in a lab because we've never seen this before in nature. These French scientists who recognized that the difference between SARS-CoV-1 in 2003 and this 2020 SARS-CoV-2 virus were these spike proteins. So they took the spike proteins and ran the DNA testing, and they wanted to know what's the gene sequence identical to. And oh my God, the DNA experts published it was identical to two things. The king cobra's venom protein called cobratoxin, and then a second venom called bungarotoxin, which is from the Asian crate snake. Then they said it also has similarities to parts of the rabies virus gene sequence also. So now you've got two different countries with DNA experts confirming the spike proteins of COVIDs are venoms. If you want to know why I've assembled this group of people to come and join me for the Healing for the Ages conference, not because it was comfortable for them to talk about venoms being a part of COVID. There's actually one guy over here, Dr. Ely, who for a whole year was like, I ain't even talking about that. I don't want to look at it. I'm not listening to that crap. Until he, on his own, decided to prove me wrong and went into his own research and was able to find the exact same publications, not by me, but by geneticists in multiple countries in the first 6 months of COVID. So, going forward, what we now know, the NIH on their website right now, go to nih.gov if you'd like, and type in how we manufactured the COVID-19 vaccines. There's a pamphlet for all of you at home to read. I just found this like 3 weeks ago. And it explains in two pamphlets, we did not put SARS-CoV-2 virus inside of any of our COVID-19 mRNA vaccines. This is why we can say confidently, no one can get COVID from our COVID-19 shots. We didn't put the virus in there. Okay, this should be quite alarming for all of you. They told you, in order to stop the spread of COVID, you can't go to work. You can't get on a plane. You can't hug your grandparents in a nursing home unless you get the COVID-19 shot to protect and prevent for worse outcomes the SARS-CoV-2 virus infection. Yet they published, they didn't put the virus in there. However, in that pamphlet, they do tell you what they put in there. So we're going to drive home for you that we know exactly what we're talking about, and we're going to teach you exactly the all-natural substances and solutions God put on the earth that actually solve this problem for everybody who was injected. That's 5 billion of you at home. The NIH has on their website right now, we chose as our vaccine candidate, I quote, the spike protein gene, and we suspended it after synthetically manufacturing it in what is called a DNA plasmid. And they show a circular DNA structure that they make in a lab, and inserted the spike protein gene inside of it. Well, team, what did geneticists determine was the spike protein genes of COVID? They were venoms of cobra venom. It's called cobra toxin, and then a substance called bungarotoxin from the crate snake. This is important. Inside the COVID-19 mRNA shots, they said we didn't put any of the virus. We only put these DNA plasmids with the spike protein gene inside of them. This is important. I want to put up the first slide because this team didn't even realize, including myself, how long they've known they can actually make a mammal like you and I, how they can make a mammal manufacture king cobra venom inside of your body, inside of your cells. And I only found this study about 4 weeks ago. And we're going to put up the slides here. Let's put up the first slide. So it's titled, Can a Mammalian Cell Produce Cobra Toxin? Which is a part of the king cobra venom, determined to be the spike protein of COVID by geneticists, not medical doctors, not chiropractors, definitely not podcasters, geneticists. All right, we're going to trust the DNA experts that they didn't screw up and didn't know how to do their job. I think they got it right the first 3 times in the first 6 months of COVID. All right, so next slide. This is the title of the article, and I'll read it out loud. It actually reads, in vivo. What does in vivo mean, Dr. Group?
In the body.
In the body. In vivo, in the body, trans-splicing of 2 DNA plasmids called 5' and 3' segments of what is called pre-mRNA. Not mRNA, pre-mRNA. Directed by corresponding DNA sequences delivered by what they call gene transfer. Any of you heard of the term mRNA gene therapy in pertaining to these shots they call COVID-19 mRNA shots? All right, the NIH just said they put DNA plasmids with the spike protein gene inside of it. The spike protein gene was found to be bungarotoxin and cobra toxin. This paper is from the year 2003, 20 years ago. All right, so let's go, next slide. All right, these studies were supported in part, I love this by the way, by the Will Rogers Fund. What? And the Malcolm Hewitt Weiner Foundation, and in part by the NIH and their funding 20 years ago. Next slide. We have developed a new paradigm of in-the-body gene transfer termed segmental trans-splicing, putting together 2 things, in which individual donor and acceptor DNA sequences, you guys don't have to memorize this, remember you're going to get all this at home, delivered in vitro, which means in a lab, and in vivo, in the mammal's body, your mammals by the way, generate pre-mRNAs with 2 primed DNA plasmid segments, we'll explain what those are in a second, respectively. I want to go to the next slide. Cells or animals receiving both donor and acceptor plasmids. Plasmids, 2 plasmids, DNA rings, with different parts of half of the king cobra toxin, half in one, half in the other, 2 plasmids. But neither plasmid alone. They actually made 2 plasmids, DNA plasmids, inserted cobra toxin into it, took those individual half pieces of cobra toxin inside of a plasmid and injected that into animals by itself. And what they profess in this paper is they wanted to see if we give them half of the cobra toxin in this plasmid and inject it into these animals, will their body manufacture the complete king cobra venom cobra toxin mRNA? Then they took the other plasmid with the other half of the cobra toxin, injected that into a mammal, and wanted to know, will that animal take that part of the cobra toxin inside of a plasmid and get it absorbed in their cells and then spit out the full king cobra venom cobra toxin mRNA. That's why in this paper they call them 2 plasmids pre-mRNA. Okay? So they inject this into them. 2 plasmids, and it says, but neither plasmid alone, when they injected the individuals into the animals, nothing happened. The animals with 1 plasmid, 1 part of the cobra toxin, when they injected that in the animals, nothing happened. They didn't die, they didn't produce any mRNA, nothing happened. They pooped it out in the toilet. Got it? When they actually took both, we'll keep going, when they actually gave them both donor and acceptor plasmids, the 2 of them, as you read here, but neither plasmid alone, it yielded. Yielded means produced and manufactured. Sorry, I have to look past group here. Yielded with PCR product 20 years ago, with the correct sequence, the 2 plasmids injected into the animals separately. produced mature alpha cobra toxin mRNA.
Hold on a second. Do y'all get that?
Yes.
Do y'all, I mean, honestly, do you get that?
Hold up, wait, watch what happened. So they produce cobra toxin inside the cell of the mammal. The mammal draws in these DNA plasmids, and then inside the mammal cells, it transplices the two pieces of cobra toxin, creates the mature alpha cobra toxin, spike protein gene, mRNA, releases it from the cell into the animal's interstitial fluid and bloodstream, and watch what happened to them. Suggesting that transplicing had occurred, mice receiving the intravenous administration of both donor and acceptor plasmids, but not either plasmid alone, died within 6 hours. Keep going. Next slide. With donor plasmid only, we observed only mRNA with the donor sequence. Remember, I said they put in one plasmid, one. You only got the plasmid with that one piece of the cobra toxin. With the acceptor plasmid only, we observed only mRNA with the acceptor sequence. In contrast, animals who received the donor and acceptor plasmids had mRNA in liver, corresponding to donor, acceptor, and full-length alpha cobra toxin mRNA. 80% of all the animals died within 6 minutes. That means the mammals produced cobra venom by putting two cobra toxin pieces together, spit it out, killed the animals. 100% of all of them died within 6 hours. The moment I sent this to this group of individuals, this is not a joke, I sent it to them, their immediate response was, oh my God, does this explain for the first time in history that people were not considered vaccinated unless they got 2 shots? Did they introduce in the first shot DNA plasmids of one of these, 2 weeks later inject the other one? And my immediate reaction to that question was, do you guys know how many people died and after which shot? 90% of all the people that died from any of the COVID-19 mRNA shots died within 48 hours after the second shot. And that fits the same time frame you see here. Now I want to go to the next slide because I'm suspicious that the next slide might be what I want to show. Okay. All right. The very next thought was this from Dr. Henry Ely. Ely, go. I want you to talk about this. Because once you know and understand you can get a mammal to make venom in their body, in their cells, 20 years ago, and you start to realize, oh my God, is it true? Maybe that's why they had to have the 2 shots. And then why the injuries were so pronounced immediately after the second one. Ely, what did you look up and what did you find?
This is crazy, right? So here's the question. If you had to get 2 shots and it's the same stuff in the vial, why in the world would you put a different color cap on the vials? Why would you have a purple and a gray cap? And this is Pfizer. This is directly from Pfizer BioNTech COVID-19 vaccine insert. Primary series, look what it says here, for 12 years of age and older. The Pfizer didn't even approve it for anyone under 12 years of age, but our FDA did. Look at this. The purple cap must be diluted prior to use. And then for your second shot, you get the gray cap. If it's the same stuff, why do you need a color coding? Does that make sense, everybody?
So when you would show up to get your shots, they would ask you, is this your first shot? Yes, good. We'll grab this color cap. This is for the first one. Boom. Yeah. Come in. Is this your first or second shot? It's my second shot. Oh, we got to grab this color cap for the second shot. Yeah. Why else would you need to designate them by color if they're the exact same shot? Now, this is important for everybody at home. I mentioned a ton of a myriad of symptoms that people can live with. Every single symptom of COVID-19, from the symptoms of COVID, the acute one, the long-term ones, what they're calling now post-COVID syndrome, long haulers syndrome, with symptoms, a collection of 21 of them right now, people living with a year and a half or 2 years post having a mild case of COVID. And every single COVID-19 vaccine injury, regardless if it was an mRNA shot or not, can be explained with cobra toxin, bungaroo toxin venom from these snakes, and the 36 other venoms found in COVID-19 patients by Dr. Carlos Bragna in Italy. He confirmed in every COVID-19 patient in Italy in 2020, in 2021, they had a collection in their blood and in their feces of over 36 different animal venoms, including 20 snakes venoms and a bunch of cobra venoms called cobra toxins, crate venoms, bungaro toxins, and also 15 different cone snails out of the ocean floor. Their venoms were in the patient's bodies, only in COVID-19 positive patients, none in the control group who did not test positive for COVID, and also a starfish from the ocean floor called crown of thorns starfish venom.
Right.
Now, either all the people on land, seriously, either all the people on land in the multiple cities in Italy they collected these patients from, this is published, by the way, October 2021 in medical journals, right now online. Either all these people from multiple cities together went snorkeling out in the ocean, swam to the bottom, and had these little cone snails inject them all with 15 different venoms, got bit by a starfish called the crown of thorns starfish, that only one. And then they had to go to Malaysia to get bit by the crate snake. They had to go to China to get bit by the king cobra that they found. Then they had to go to Uruguay and get bit by the coral snake venom that they found there. This is not a joke. Then they had to go to Taiwan to get bit by the taipan. Then they had to go to Japan to get bit by the habu snake. Then they had to go to India and get bit by the eastern brown snake, by the way, which caused all of the blood clotting effects of COVID-19 was their venom from the eastern brown snake. It's called E-carin, is the protein. All right. They know how to make the human body, a mammal, make venom in your cells using DNA plasmids with spike protein genes in it. And that's exactly what the NIH published is in these COVID-19 shots. And not a single human have I heard outside of this group talking about plasmids with instruction of venom inserted into them. And these plasmids have been designed for over 72 years as a concept. And it's exactly how they created the COVID-19 narrative, how you got sick. They can distribute these plasmids through air, water, food, inject them into you as vaccines. And this is how they're going to create every single pandemic coming right now, including the Marburg virus you're hearing hints at, the smallpox virus that's coming up next, the West Nile virus from these GMO mosquitoes. Just so you know, they are all DNA plasmid injected with venoms they're going to call viruses. Oh, by the way, did you know that the Latin definition of the word virus
What is that?
means venom? Oh, and what language does the pharmaceutical industry love to hold on to that nobody else on the world uses?
Latin?
Latin. So just remember,
Curious.
this is actually what you're going to learn here at the Healing of the Ages. And the great news is there's actually substances on Earth, in plants and in the dirt and in herbs that neutralize cobra venom in a human and also destroy plasmids when they're entered into your body, regardless of how it gets there. That's what you're going to learn at Healing for the Ages. And I will be presenting that information Saturday, which will be September 9th. All right, now it's time to turn this over to Dr. Group.
Can we give it up?
All right.
Online, we love you. Can we give it up a little bit?
Give it up, Ambassador.
That's one research paper. One.
Oh,
What?
okay.
We're in here all night.
Oh, yes. As long
So
as that screen works.
it gets deeper than that.
It does.
So first off, we're here really because we love everybody, and we're looking at this from a multifaceted approach. So if you look at the spoke and hub of a bicycle tire, let's say, and you have the hub there, that's the person. You have so many different vectors. And a lot of the individuals and the scientists and even ourselves at certain times have focused on one subject. And this is exactly the way that we're engineered from birth, is through the education system, to be an expert in one subject, but not knowledgeable of many subjects. So we have to open up our minds and explore what are these other vectors that they're using, and what is the main goal of these dark forces that we're dealing with on the planet right now, going through this Great Awakening. And I'll say that because there's also good forces. Like, I don't want everybody to think there's just the bad forces out there, because that's why we're here. That's why everybody's here. That's why the great scientists, the great doctors are out there working, around the clock to try to fix this. We've been studying this for 25 years, and I've come to the conclusion, or just ask yourself this question: what if my whole life has been engineered and I don't even know it? They have engineered the education system. They've engineered the political system. They've engineered race. They've engineered the food system. They've engineered the water. They've engineered the weather. They've engineered synthetic biology. What we have to understand, and what my talk is going to be on, is something the first day which we don't talk enough about, and that's the anatomy of the energy that we are. Ely has the Energetic Health Institute. Bryan is one of the top AK muscle testers in the world. Jan is around energy from the bees all the time. We all have a aura around us. We are, just like Tesla and Einstein said, we are energy and vibration. And ultimately, this multi-vectored attack on us, whether it's they've gotten us addicted to sugar, they've gotten us addicted to alcohol, they've gotten us addicted to social media, they've gotten us looking outside of ourselves instead of focusing and working on the energy and the vibrations of us becoming enlightened beings on this planet. It's the suppression of our own God-given will to become enlightened beings. So as we're looking at all of these different vectors, what I'm going to be covering is what the true plan is, which is a transhumanism agenda. This is a word that's not talked about a lot because it's kind of a scary word, but it is a hacking, which they've been doing for quite some time, of our bioenergetic system. And by the way, all of the things really can do one of two things. They can lower our vibrations or raise our vibrations. Cooked, dead, processed food lowers our vibrations. Negative emotions, fear, anger, depression lower our vibrations. Watching social media or watching mainstream media lowers our vibrations. And everybody at this point in time, their plan has come together. And they're using what they've convinced us all are cell phone towers, which are actually directed energy weapons, including the Wi-Fi routers inside your home that can be used for different types of frequency modulation, to be able to utilize all of these vectors to try to transform us into basically, and I know this sounds crazy, but we're going to be showing you all this research because you have to look at all of these different things to be able to piece all this together. Why are they using snake DNA? We're gonna reveal that this weekend at the conference. But did you know that roughly 97% of the cells in your body are non-human? Do you know that? Roughly 97 to 98% of the cells in your body are non-human. They are not only targeting the mammalian cells, but they're targeting all of the parasites in our body, which most of those have been genetically engineered, Morgellons and Lyme and malaria and all these other different types of, bird flu and all these other organisms that they've been genetically engineering. We are riddled because what do people do during the lockdown? They eat a bunch of sugar and they drink a bunch of alcohol. Well, guess what feeds the parasites? E. coli, candida, yeast, it's all, you're feeding these parasites in your body. By the way, a parasite is anything harmful that lives off of a host mechanism. That's going to be your mold, your viruses, or your foreign proteins, which they call viruses. That's going to be bacteria. It's going to be synthetic parasites now, which, if you would have asked me about synthetic parasites like 10 years ago, I was like, I don't even know what you're talking about. But now we're coming into the age of this cyborg transformation of creating this controllable human. And this is all documented. This has been going on since the 1960s, actually. They call it the wide body area network. You can look it up online, the W band. They purposely do not teach anybody in school that they are an energetic being, like Tesla and Einstein. Were they trying to tell us something when they both said everything is in energy and vibration? So they're basically laughing at us to a certain degree when we're out here talking about the spike protein, because that's just one tiny vector. But if you really trace it back, they're using the parasites because parasites are great carriers, and worms can have millions of bacteria and fungus. And because your body is made up of 98% non-human cells, the cells of the parasites are becoming a massive manufacturing facilities for these venomous spike glycoproteins. So this is a multi-vectored approach with the food, the water, the air that you're breathing. And what we would like to do is to give people the confidence, because sometimes when you're under stress or anxiety, it's usually from not knowing. People didn't know they were going to die of COVID, so they were in an intense state of fear and anxiety, which, by the way, lowers the vibrations by 50% in your body instantaneously.
Can I give them an example real quick?
Yes.
Of the unknown? You guys ever called somebody you know and love and they didn't answer the phone?
Yeah.
And then you called them again, they didn't answer the phone?
Yeah.
And you called them again, they didn't answer the phone? What did your brain start telling you? Something's wrong. Why? Because you had no knowing. You had no idea what was on the other side. So most of the time, us not knowing something creates the most fear, anxiety, panic, and confusion. This is just an example. We've all lived through it. I do want you to explain something real quick before you keep going, though.
Okay, good.
Because I can see all the brows in here. When you say 98% of our cells could be non-human, what is it? Alien? What is
No,
it?
just bacterial cells, mycoplasmal cells. Just
wanted to clarify that for y'all.
So one of your human
Because
cells.
they don't know. I knew they were gonna make up all kinds of
That's
stuff.
some crazy stuff with that trans person too.
And these, basically, the NIH has actually come out and said that you have about 90% of the cells in your body come from your microbiome, which is mostly bacteria and yeast.
There you go.
However, there's no studies that take into account all of these synthetic or all the man-made bioweapon type parasites that they've done, all the liver flukes, all the tapeworms, all the flatworms, all the pinworms, all the mycoplasms that live everywhere in your body. It's not just the microbiome. So how they can literally turn these on and off with different types of frequency bursts as well, which we're going to get into about how. I have some slides right here. So we're looking at, and by the way, we're calling out also for help from all the scientists. This is an effort that we want everybody to review all of our research. We're going to be publishing all this and all of these vectors of what are the best natural ways to get rid of the parasites in your body? And you have to target the yeast, the fungus, the mold, and all of these things. What is the best way to neutralize these venoms that are being produced? What is the best way to actually repair your DNA from any type of damage that has been caused from your DNA? What is the best way to protect yourself and your loved ones from the electromagnetic frequencies that they're using? What can we do about all that? So you're going to get all of these solutions, and there's probably many more solutions out there too. At this point in time, we just want to bring this to people to show that there are so many different things and so many different ways that they're using as vectors to initiate this plan. And that's not even counting what we're going to talk about with the nanobot technology, the quantum dot technology, the self-replicating hydrogel technology, the synthetic hydrogel technology. I mean, there's literally thousands of papers and research articles on all of this information. And there's not a lot of people that are talking about the DARPA-funded studies and the DoD and the Army military studies that they've been hacking into our biofields for a long time. That's our energy. They want to take our energy away from us. So we're going to put those pieces together for everybody. But at the same time, that doesn't mean that they're going to accomplish their mission either. Okay, because like I said earlier, we have the good guys and we have the bad guys. And we all believe that we're going through this great awakening process right now and calling on more people to send us information and to do the testing and to get more information out there.
All right, you got 5 minutes to go
I've
through your
got
slides.
5 minutes. Okay, so,
I'll give you 5.
when we're talking about EMFs, this is radio frequency, extremely low frequency emissions, and electromagnetic frequency emissions that I feel during the last pandemic. I don't know if you guys saw the whole meeting with all of the telecommunications heads and how they were going to fast track these towers everywhere, which I believe have the ability to go from megahertz range to gigahertz to terahertz, and potentially even do radio frequencies and microwave as well. So this is the broad spectrum of the harmful. Because we have positive, like the solfeggio tones and good music, like Mozart and all that. Those are really good, powerful frequencies too that can build up your energy levels in your system. So right here we had from the NIH, which is the preliminary results on the non-thermal effects of 200 to 350 gigahertz radiation on the growth rate of S. cerevisiae, which is a yeast, actually brewer's yeast, which by the way is allowed in you can go buy it in the grocery stores, and it's even the engineered form of this yeast is allowed at up to 10% of feed for cattle as well. So they can spread this everywhere. And they've already, by the way, been spreading the quantum dots and all this stuff everywhere as well. Okay, next slide. You can see here that the ionizing radiation here, that as the yeast were exposed to these electromagnetic fields, go to the next one, statistically significant enhanced growth rate was observed at 341 gigahertz. Growth rate was assessed every 30 minutes via time-lapse photography. So you can go to the next slide. But anyway, we're going to talk about all these different microbes that they've been using inside your system as delivery vectors. They've been changing the genetic codes of all these organisms in your body as well. And these organisms become more resilient to damage because most people's body fluids are alkaline. They're toxic. They're positively charged instead of negatively charged. Basically, what this comes down to is they're using lots of positively charged stuff, even in their hydrogels. And what we need is negatively charged stuff. But people aren't eating enough organic, fresh, raw fruits and vegetables, and they're not drinking the right type of water and doing the things that they need to do to raise their vibrational frequencies. E. coli is what they use actually with plasmids and genetic modification. Right now, they like P. pastoris, but they've already been working on synthetic, not even living cells. They're already doing stuff with synthetic cells at this point in time. So glucose, by the way, is the main food for E. coli. So if you go to the next slide. So when E. coli was submitted to a 5G frequency generated by alternate 60 hertz voltage source, the differences on growth in glucose consumed in control and exposed groups were evaluated using the U-test. There was significant difference in glucose consummation and growth in E. coli after 8 hours of exposure to electromagnetic fields. So this is, your cell phones, your Dr. Ely's going to be talking about the blue light, how the blue light activates some of these things, and that's why you see if you type in quantum dots on Google, it's going to say quantum dots are liquid semiconductors that emit mostly blue light because they're getting it in. They need it inside. They're giving it from the outside, and they're also giving it to you from the inside as well. All right, next slide. Exposure to 50 hertz. I mean, there's thousands of these studies out there. Just like this, I had a hard time just picking one or two when I was going through all this. But this is really where everything is leading to, this transhumanism agenda. They know that a lot of people are not going to be able to survive this if their plan goes through, I should say, to survive this, and that's what they term the kill-off 60-70% of the population, because they're already sick and diseased, and those are the useless eaters, which they call people. So this is 50, most people have Epstein-Barr, most people have Lyme, they don't even know it. 50 hertz electromagnetic field induces activation of the Epstein-Barr virus genome. And by the way, I've got studies from the DoD that shows that they can fly drones over and target an individual specifically with different types of frequencies, and they can even read, and they have been reading your energy fields for quite some time. So this is something that is, to me, there's a big diversion when they have you say, look over here at these shots. I mean, yes, that's very, very damaging, but they're attacking the mammalian cells as well as all the parasitical cells, and ultimately they want the group of people that can survive and become basically synthetic humans. And we're going to go over that in a little bit more detail and how we can avoid that and how we can dissolve the nanobots and how we can get rid of all that stuff. Next slide. So a human lymphoid cell line infected with the EBG genome to 50 hertz EMF resulted in an increased number of cells expressing the virus early antigens. So they've been working on this technology, like Dr. Ely talks about, since they brought all the Nazi scientists over in the late 40s, and really they started all these black op projects in the 1960s, and everything was engineered. Every single part of your life was engineered, from the time that you were born in the hospital, to the injections you received, to the type of schooling you received, to what you saw on TV, to getting you hooked on fast food, to getting you full of parasites, to, they had all of this stuff completely mapped out and engineered all the way through the pandemic. And the last piece of the puzzle is to finish the engineering of you as a human, from a regular human to a synthetic human.
Great
You
job.
got a couple more slides. No, hold on. He hasn't
Okay.
given his punchline yet.
All right, so
what's
go
All right,
to
you
the
got
next
30
slide.
seconds for a punchline.
All right, go to the snake venom peptides. There's a reason why, by the way, they're using these. I will not reveal that right now.
Okay, well then you don't have a punchline.
Yeah,
Okay. All right, go to the next slide. Let me just see. Okay.
This technology with the telecommunications is linked to the venoms.
Yeah,
Right.
totally.
That's why they're using them in
Well-
COVID and in the vaccines.
Well, he was
because of
trying
the
to
tech.
save it, man.
I know.
Right,
You don't
he's
know
trying
yet.
to save
I didn't tell him what it
it.
is. You'll see.
Okay.
They're related.
Okay.
Well, because it's like, well, why are they using the snake venoms?
We know.
Yeah,
I'm
why?
going to
You'll
show
learn why.
you a video on Saturday of a temple in India that's 1,200 years old that has literally pictures carved into the temple of genetically engineering a human with snake genes 1,200 years ago, where the human is turning into the snake, and there's statues in the temple of a part human, part reptile. And the tour guide who's in the temple actually says, are they trying to warn the future generations of what happens when you start messing around with genetic engineering? And
You got 2 more slides.
that's what.
No,
that's
you
what-
don't.
Yeah,
No,
no,
well,
yeah,
that's-
go do it again.
All right, well, you only got 5 minutes. easy
Okay,
with your
all right.
I gotta cut my time, right?
I give you my time. I give you my
So,
time.
all right, I'll run through this really fast. Okay, so why snakes? Because they know, and if you your own research, you will see that our DNA, human DNA, is very sensitive. Masaru Emoto said this when he was doing research on water. He said the worst thing that I've ever come across is somebody being continually exposed to harmful EMFs, because the water never has a chance to restructure itself back into a crystalline structure so we can communicate. All living beings are at least 75% water. Our human DNA cannot handle the transformation from human to synthetic cyborg human, not even human at that point. So they have to strengthen the DNA, and that's why they're using the snake DNA to infect all the parasites in your body and all your human cells in your body. Because As you can see here, they used snakes when they went into Fukushima. And in this study right here, they found few adverse health effects exposed to levels of radiation. The two things they found that were resistant to the radiation were wild boars and snakes. But the snake genes have a, I can't believe how much, like everything that all the research I do, it's like ends up coming back to snakes.
It keeps coming
It's
back
like
to
the
snakes.
World Health Organization target over the world.
I'm gonna show that one in a second.
With the
Yeah,
snake.
yeah,
I mean,
okay,
it's,
all
it's
right.
crazy.
All right, so are there solutions to all this group? No,
There
we're gonna
are
get-
solutions. And here's really what we want everybody to come away with. Your future is one decision away. That's it. All you have to do is make one decision, and that's to change your life. You cannot continue to poison yourself every day. You cannot continue to be wrapped up in social media and all that other stuff. We are going to provide solutions. Actually, a lot of the solutions we're going to provide are from nature and for free. Just so everybody knows, we're not here to promote any of our products or to try to sell you a bunch of programs or anything like that. We're here to teach you the vectors that they're using and what we've found in our research, which our research is going to continue and continue, that we feel confident in to address each one of those vectors that they're using that we're going to identify.
Awesome.
All right, next slide. Next slide. Okay.
All right.
That's it.
Can you give it up for Dr. Ardis?
So that's a tiny amount. That's a tiny amount of information that we're going to be talking about. But putting it all together.
And man, I thought snake venom was controversial.
Yeah, right.
Yeah, this is the venom doctor. I'm the urine doctor. This is the health engineer.
the Nazi guy, right? The guy
And
that's about the Nazis.
Jana is the bee queen.
Yeah.
So this is
Yeah.
definitely a controversial but loving team that Dr.
Yeah.
Ardis put together that we can we've dedicated so much time and effort to just bring everybody this information and in a way to teach everybody to feel confident that they have the power which God has given us all the most powerful healing mechanism in the world, and it's inside of us. It's not outside,
Amen.
it's inside of us. It's our self-healing mechanism, and we're going to teach you how to reactivate your body's self-healing
And I don't know about y'all at home, but I actually am super thrilled to learn from each of you at the
Yeah.
conference.
Yep.
And see your presentations
Yep.
because we learn from each other every single day, and that's
We
going to be
do.
what's exciting for us. It's going to be very cool. All right, Dr. Henry Ely,
Yeah.
it is time to turn this over to you. You got 7 minutes.
Oh, great, thanks.
I'll share
7
some of
minutes.
my time with you.
Listen, I'm going to tell it to you like this. When we win, we're going to actually publish our huge long text thread. So you can see it probably be like longer than, War and Peace and everything, because we get at this every day. What you're seeing right now is literally less than 0.1%
Yeah.
of what we can bring to the table. And that's why what Dr. Group was saying, we're doing this because this conversation has to start now. They are so much further along than any of us suspected just 2 years ago, right? Yeah. If you would have told me 2 years ago, this is what they were capable of trans-splicing and having a mammalian cell make cobra venom, y'all? Come on. This is crazy, right? And then you start talking about them trying to get the snake DNA embedded into our DNA so it can withstand their Internet of all things, this frequency and this 5G. These are crazy people out here, okay? They're Can we all just agree? These are some crazy ass people.
Psychopaths.
All right? So let me tell you, first of all, I'm the only dude up here that on Friday and Saturday, I got a parental warning on my front screen because God has given me the permission to let the expletives fly, and I got a lot of shit to say. So we're going to have some fun this weekend. We're going to be some real people having some adult conversations, okay? So, my goodness. Dr. Group was talking about something so important, yeast, right? Yeast. It's in everything now. It's in beverages. It's in potato chips. It's in soups. And you start asking yourself, why is yeast everywhere? Well, because it's genetically modified, because it's a delivery system. And that's what you have to get your mindset in so we can simplify this. What is the delivery system? If you understand the delivery systems, now you can take back control. And what we're after is empowering all of you to something very, very simple. Building a lifestyle. Building a lifestyle where you don't have to worry about these psychopaths very much. You have to worry about them a little bit, but you don't have to worry about them very much, okay? So one of the fun things on Saturday I'm going to be doing is I'm going to show you a home setup for how to purify your water and structure it, like Dr. Group was talking about. And we're going to show you how to do that. It's very inexpensive, and here's the cool part. The water is their major delivery system. You get control of your water, and you knock out at least 50% of their delivery systems. At least. If you say no to these damn shots, please tell me y'all online are not taking another damn shot. Please, with God above, just please tell me if you're online, you are not taking a, have we, oh, wait till
I promise I won't take one either.
Yeah, I know you.
After tonight.
I am about to break your idea of what a vaccine is on Friday. I've already been working on that. I haven't got my presentations done yet, but they'll be done before, okay? Let's go to the, so let me read a question here. Hold on, Jordan. How long can a weaponized microbiome produce spike glycoprotein?
Good question.
Good question, right?
Yep.
Audience, and you online, tell us, type your answer in. How long do you think a microbiome, a cell, a bacteria, or a yeast, how long can it produce spike glycoprotein? Give me some answers in the crowd. Come on, participate with us.
12 months.
12 months.
Forever.
Forever. Hold on, say it the right way.
Forever.
That's Silk, y'all. She's saying, she's saying, forever. That's what I'm talking. That's where she is right there. Forever. And guess who's right? She's right. They did something very interesting. They put on the plasmid something called a promoter. And when they put a promoter on there, you have to ask yourself, what is it promoting? Well, it's promoting the production of spike glycoprotein. Venom. It's promoting the production of venom. Now, there's a word I want you to say with me and help me out. Some of these words are hard. We've learned some words that I never thought existed, right? And you're going to learn some with us too.
Yeah.
So check this out. The word is constitutive. Can you get that out of your mouth? Because it messes with me every time. I'm like, constitutive. That's a hard word to say. You know, can you say it with me? Constitutive?
Constipated.
Constipated. Leave it to Brian, right? Constipated.
He's definitely saying constipated.
So now,
I'm feeling constipated trying to say that.
all right. So if you hear a loud roar in the back when Brian's on, it's me. I'm going to be rolling when this dude goes on stage. He cracks me up. So, all right. So yes, constipated, right? What it means is that it's always turned on. Okay? And if it's always turned on, that means it's always making the spike glycoprotein. It's always making it, right? So here's the deal. Why would you do that? Why would you do that? Right? That doesn't make sense. This was supposed to be a short-term thing, give it a little bit of a challenge, and then your immune system picks up the challenge, and then you have immunity. That's what they say, right? It's supposed to be a little short thing. No, they didn't design it like that. But it gets a little crazier. Because on this plasmid, right, these mother beep. I'm gonna have to beat myself on stage tomorrow. I know it. Listen.
Mofos.
Listen. I want to drop it like the way I want to drop it. You know. No, all right, I'll go with that today. Mofos. We'll be in the world. I want to say some other shit, but okay. So they have on these plasmids multiple promoters, and this was one of my questions to all these life sciences dudes. Right? I was like, okay, I see the promoter for the spike glycoprotein, or at least half of it.
Right.
Why you got 3 or 4 of the promoters on this plasmid? Can you tell me what they do? And I'm talking to these brainiacs, right? And they're like, I'm like, wait, hold on. Y'all the best in our movement. I need y'all to tell me what this is. They couldn't tell me. So we started doing a little bit more digging. And what came up was, how many of y'all heard of those GM mosquitoes that Bill Gates been talking about, right? You see how I'm talking about them now, right? I did not slip my tongue. You all caught that, right? Okay. Those are made by a technology called CRISPR gene drive. And we're going to talk about this on Saturday because they have proof of concept now that they can target women. All right? Now, I don't know about you. I'm serious about protecting our women, and I know everybody else up here is too. They can specifically target our women to make sure that every single woman is infertile within 3 generations. They have proof of concept. This isn't 10, 20 years into the future. This is today. Today. So why did we push forward and why did Dr. Ardis bring us all together and say we got to do this now? Because no one is talking about this in our movement. We're talking about masks still. We're having arguments about masks. Are you kidding me? They're trying to kill us and you want to talk about a mask? Just don't put the damn thing on. I mean, that's an easy fix. Let's get to the real conversation. So that's what we're doing here. So how long can a weaponized microbiome produce spike glycoprotein? Here's the punchline for you. Our mammalian cells weren't the original target. The original target was the 400 trillion microorganisms within us to be incessantly producing spike glycoprotein, venoms. Let it sink in for a second. When somebody is trying to kill you, you better believe them, because they're trying to kill you. Now, I can't wave a bigger flag than that, because I hear everybody in there, whoa, that's kind of heavy, Dr. Reyes. I don't know what you do. Yeah, it's kind of heavy, right? But that's where God taps you on the shoulder and says, I'm going to give you Dr. Janet Schmidt. Because she's going to lighten it all up.
They actually put in papers that we can create with this gene drive an extinction event for the species.
Yes.
In 3 generations.
They
Extinction event.
brag about it, right?
They flat out brag about it.
Let's go into these slides. I'm going to show you some cool stuff that just came out.
And just so y'all know, every single topic we're talking about tonight are all interrelated.
Yes.
They are.
That's why we're presenting this all together.
Yeah.
The venoms, the towers, the microbiome,
The plasmids.
manufacturing
Yeah, it's all of it.
venoms
All
and
of
the plasmid. All of it is interrelated.
It's all there. It's why they put us in lockdown. Isn't it a little interesting to you? Because it's a little interesting to me. It's a little interesting to you that they said we're having the worst pandemic since the Spanish flu. They said you need to stay at home and be very afraid. Don't hug your grandmama, right?
Don't go outside.
And while we were inside, magically there are towers emitting 5G radiation, not in one state, not in one little local community so we could try it out, but in every single country in the world. If that doesn't ring an alarm, I don't know what can ring an alarm for you. So let me show you something here that's good. We got some good news.
It wasn't just for Netflix.
I thought it was at first, but then my stream sucked. So what are you going to do? So check it out. So this just dropped a couple weeks ago. There's Carlo Brogna again that has been doing, to me, clinically, the best research in the world. The best researcher in the world is on the stage. His name is Bryan Ardis.
Yeah.
Okay? The best clinical tester, the developing tests, is Carlo Brogna. All right? Now, this says detection of recombinant spike protein in the blood of individuals vaccinated against SARS-CoV-2, possible molecular mechanisms. Okay, we have been waiting on this test for 3-4 years. My number one question has been, how can we tell whether the spike glycoprotein came from the damn infection? And I'm going to say damn a lot, just get ready for it. Or from the damn shots. Okay?
Right.
We have not been able to distinguish this in over 4 years. Carlo Brogna figured out how to do it. Guess what he just proved? Let's go to the next slide. Authors declare no conflict of interest. You're going to see us share this with you. We want you to become students. We want you to become researchers. We're going to show you how to vet research. I read the title, and then I go straight down to the conflicts of interest and any funding statements. And then I make my determination. Okay? Let's go to the next slide. Mass spectrometry. Am I saying that right?
Yes.
Damn, I got lucky. Examination of biological samples was used to detect the presence of specific fragments of recombinant spike protein, and it should say spike glycoprotein, in subjects who received mRNA-based vaccines. Next slide. Although the mRNAs are different, both encode the same recombinant spike glycoprotein. This differs from the natural one by a double amino acid change at position 986. These some nerds, y'all.
Yeah.
Right?
Yeah.
And 987. The amino acids being lysine and valine are both replaced by 2 proline amino acids. Carlo Brogna figured this out. That's why you need a nerd around. They come in handy. All of us on stage are super nerds. We come in handy sometimes, okay? We might have different levels of style and different levels of tact, social tact, but we come in handy. This is a dude you need on your team. Go to the next slide. The minimum time spike was detected was 69 days after getting the damn shot, while the maximum time was 187 days. Does the CDC tell you that you could be producing spike glycoprotein indefinitely? No. What's the 3-letter stupid ass phrase that they say all the time?
Safe and effective.
Safe and effective. I'm gonna blow that up on Friday. They got a problem in me. And this is what I want y'all to know. If somebody's trying to murder you and the people you love, you better become a problem for them. All right? Can I get an amen in here?
Amen.
Okay. Can I get an amen online? Give it to them. All right. Let's go to the next slide.
Preach on, preacher.
I'm going to bring it, baby. You should never give me a mic. All right. Guess what Carlo Brogna does, right? He confirms everything Dr. Ardis, Dr. Group, me, Dr. Schmidt, and our teams have been coming to this conclusion. Dr. How do I say it? Shetty? Am I saying
Dr. Chetty.
I say Chetty,
Chetty,
but Jono keeps saying Shetty to me. And I'm like, we know, brother, we love you. You're right.
Dr. Chetty, you're right.
Okay, which
South
one
Africa.
is it? He said, yeah.
Love for South Africa.
He said it's Shetty. Is it Shetty? He said, Jono's like, no, it's Shetty. It's Shetty. He's in the other room, everybody. He's like, no, it's Shetty. Okay, so what is he saying? There's 3 hypotheses here that Carlo is making. Number 1, he's saying that it could be transcribed, the mRNA, into your mammalian cells. We know this is true by the Lund University study that came out last year, so that's true. Okay, they've already proven that. Number 2 said, it's possible that pseudouridines, somebody better turn that off. It's possible that pseudouridines at a particular sequence position, as described in this article, induce the formation of spike glycoprotein. What does that mean, everybody? Pseudouridine is the Pfizer. Pseudouridine is the Moderna. Okay? Pseudouridine is what creates massive protein synthesis errors in our bodies. It's something that is a precursor to turbo cancer. How many of you have heard of people's cancer just reigniting over the last couple years? That's pseudouridine. So that's happening too. And then we get to number 3. And what's number 3? Everything we've been saying all that other shit too, but it's all of it. When we talk about they weaponized the microbiome. Yes. That was their target. They weaponized it. Go to the next slide, Jordan.
You got 1 minute.
No, man, you ain't putting me on the clock after y'all took like 30 minutes. I'm gonna talk. No, I'm just playing with you. All right. So I got 1 slide. So
I know.
So check it out, y'all. This is the most important slide for you to get.
Please pay attention to this one slide.
Okay? We are going to hammer this home with you all over the weekend. You have a plasmid over here in the lower right-hand corner. You see at the bottom, it's an origin of replication. It means it can replicate all it wants to, self-replicate. That sucks for us. You go around, you have antibiotic resistant region. Guess what that means? Antibiotic resistance, Dr. Ardis, was that because they prescribed too many antibiotics, or was it something else?
They know they can create plasmids that can get inside a bacteria, and then when you expose that bacteria to an antibiotic, it won't die.
Yep. It gives the antibiotic resistance to the bacteria. Okay? That's what plasmid curing compounds do. They knock out that resistance. And we're going to show you that on Saturday. They knock it out. Then you look over here, there's a promoter, it's constitutive or constipated, as Dr. Ardis likes to say. And then you have your genetic payload. That's the plasmid. You take the 5G frequency, or whatever frequency, punch a bunch of holes into the bacteria, shove that plasmid in there, and now you have a weaponized bacteria. Are we all good so far? Are we good online? Yeah, we good? Somebody,
Hallelujah.
Jordan, Q with me. Thank you. We good? We good. Then say you good in the chat. Okay. Then we have a problem because now it can spread.
Yeah.
And it spreads through the process called conjugation. That's what you see up here. One bacteria that's weaponized, the green one on the left, sends out a pilus and it connects to a wild type that's healthy and good. Mm-hmm. And it can do this to yeast as well. And then it's self-replicating that plasmid. So what starts happening? Now you just weaponize that other plasmid. That other bacteria. Does that make sense? And how many times have we heard people got the shots, or especially got the adenoviral vector, the AstraZeneca or the Johnson & Johnson, right? And those people got it, and at first they were fine, right? And then what happens after a couple weeks? What happens after a couple months? They start falling off a cliff because of this process right here, bacterial conjugation, spreading those weaponized plasmids throughout the 400 trillion microorganisms that should be symbiotic with us. And now you get plausible deniability because there's incessant production of venomous spike glycoproteins. And you get plausible deniability because you get over 12,000 diseases created. And certainly nothing that we would ever make could create 12,000 different diseases, right, Mr. Pharmaceutical Man? Mm-hmm. Now, do y'all get why I call these things what they are? We have to have that courage to speak truth.
Now let's bring us home. You ready? How many of you have heard of shedding?
Boom.
Concerns with these vaccines. Do you know that when you weaponize a bacteria and make it to generate venom the way they're doing here, do you know that bacteria can be contagious? Did you know you can spread a bacteria to your loved one or to your child? Did you know this? Did you know you can actually spread fungal infections like yeast infections with someone you love or that you've ever touched? They are weaponizing and engineering bacteria and yeast, inserting these plasmids into them, getting them to manufacture venoms incessantly, in perpetuity, and then you're going to spread them to all of your loved ones. If you've ever wondered where the mystery of shedding and transmission is, it's in the bacteria and the yeast.
A 10-second kiss transfers approximately 80 million bacteria to the other person.
Oh, Jane, you're full of it. All right, give it up. Applause for Dr. Ely.
Yeah, Dr. H in the house. That was awesome.
When it gets heavy over the weekend, get ready for the entertainment when Ely sets up on stage.
Oh,
You'll
yeah.
get it.
And the
All right.
expletives fly.
All right, Dr. Ely, introduce the queen bee next to us.
We got the person, y'all, that, so you've had three dudes up here talking like we know all this stuff. And what's so cool is that we'll be talking and talking and talking, and then Dr. J will come in and just drop just some straight knowledge on you, but does it with such beauty and such grace and such, oh, you sweet little boys. Let me help you out here. So without any further ado,
Well,
Dr.
thank
Janet
you.
Schmidt, everybody.
Thank you. So thank you. I just like to offer great solutions, but these are God-given. We're all steeped and grounded in the naturals, and I love this slide that Dr. H made for me. Can solutions be found? Because most of the time, my answers are in something that the bee has made. Propolis, honey, bee pollen, and I'm going to get to that for sure. So yes, and an answer, yes, there are solutions. They can be found. And I have to tell you that, you know, we pray about our work. We pray for knowledge, we pray for wisdom, we pray for guidance, and then we research and research and research, and then we pray again and research, and wow, God shows up. It's just blown our minds over and over and over what's happened with all of us and our knowledge and our research.
And God shows up in the nature he created. That's what we
Yes.
say when we say when God shows up, there's always an answer
There's
already published
always
in
an
nature.
answer. Yes, it's absolutely
100%
incredible.
of the time. This isn't
Yeah.
Every
9
time.
out of
It's
10
blowing our
times.
minds. Yep, it blows
It
our
blows
minds.
our minds every day.
And so one of the things I'll be talking about tomorrow, I'm going to talk about your body systems, just get the bases, and then some of those signs and symptoms that something's wrong before it's a blown disease, right? You want to know how your body works, and so you can pick up those little signs and symptoms and some really clever little self-tests you can do. And then on Saturday, I'm going to talk about, of course, things that the bees make and how they're helping us, helping disarm these weapons, but also plants. I mean, there's so much healing in plants, and it's actually much easier than you could possibly imagine. So that's where I come in. So next slide. So this is one of the papers. When we say God shows up, this is pretty cool because one of the answers to the weaponized bacteria is glucose oxidase. So what is glucose oxidase? It is an enzyme that catalyzes glucose with oxygen to make hydrogen peroxide. Hydrogen peroxide, we may know, is antibacterial. So why is that important? Because hydrogen peroxide, this is amazing, actually destroys the lipid cell envelope or cell wall around these weaponized gram-positive bacteria. Amazing. They destroy them. So that's why this is very important. So we're like, okay, where's glucose oxidase? Well, I know it because bees make it. So that I immediately like, oh yeah, bees make that. That's
So the
how
three
they
dudes are freaking out about this E. coli bacteria weaponized stuff. And Jenna's like, wait.
I got you, yeah. So I was really excited because I knew that word, I knew that the bees made it, and that's very special. But this article was new to me. This was really interesting because there's mechanisms in noble metals, namely gold, that have this glucose oxidase mechanism. It's really amazing. Next slide. Yep, no competing interests. Next slide, that's important. We thousands and thousands of research papers, thousands. And we don't just take just because it says it for a fact, right? So we're looking, who paid for this article? Who are the people? What else are they doing? Because it may not be with ethical background. So we really research the articles before we hold on to them. So this one was great. I'm going to put my glasses on so I can really see. All right, so this one we found that gold is like natural glucose oxidation. Pretty amazing. And it's a two-step process. One is the dehydrogenation of glucose, and then the second step, is a reduction of oxygen to make hydrogen peroxide. So we were really excited to see that these nanoparticles of gold can do this. And these two are working on a pretty amazing project that will help us with that. So I won't give that away. Next slide. Okay, so the noble metals also have been found to mimic various enzymatic actions, and this is electron transfer reactions. And there's two in this case. Cytochrome C and coenzymes. And so what are they and why does it matter? The cytochrome C is amazing because it's known for its function with ATP synthesis. This is the energy of the mitochondria, the energy of the cell. This is really important because this is a secondary way we can help our cells get that energy that they need to kick off these bioweapons. Okay, next slide.
What do they call glucose oxidase? What's the term?
Oh, I'm sorry, I missed that. So good. Okay, so is it on the next slide? Okay, yeah. Oh, here it is. Okay, so thank you. So glucose oxidase, we're looking at, and I never knew that the scientific abbreviation is GOD. You could have wiped us up off the floor when we all like, what? Oh, this is better than we could have ever imagined. So thank you, God. Thank you for glucose oxidase. Thank you for gold. And next slide. Let's see if it's the one I think it is. Oh, that's not the one.
Oh.
Okay.
No, I sent it over.
Okay.
And I redid it.
All right. It's okay.
That's not on me.
That's not on it. So
Just talk about those two.
Yeah. Before we go to that, the next slides were some studies that I researched with bee pollen and bee bread. Bee pollen is the pollen that the bees gather from the flowers. They put their enzyme, glucose oxidase, in the bee pollen and they bring it back to the hive. So this shows the activities of bee pollen with glucose oxidase. Those were the research papers. And the other one was about glucose oxidase in comparison to other enzymes. They call it the Ferrari of enzymes because it's really rapid in its mechanisms and it's incredibly stable. So this is a really good solution for us. And it's nice to know that we don't have to just use the bee products, but we can also use gold. Yeah. And there's several others. So this is great. Thank you, Dr. Ely, for putting this together. So no oxidase means there's not going to be any hydrogen peroxide. And without the hydrogen peroxide, these bad guys are able to continue. So that layer around the weaponized bacteria stays without the hydrogen peroxide. But, next slide. When glucose oxidase, or God, turns the hydrogen peroxide back on, we can defeat them. It renders them useless and thus harmless to our bodies. Pretty exciting. Yeah, pretty exciting. I mean, I got goosebumps when I saw this slide. I was like, yes. I mean, this is big. This is huge. Finding ways to stop this from happening and stop it from continuing. We've been chasing the spike glycoprotein for too long. And to me, it's just a pharmaceutical model, chasing the symptom, something, a pill for every ill. But instead, we're getting to the root of the problem. And that's what we do. And I love these guys. It's their heart, too, for healing and helping people. So when I thought, how are we going to figure out how this is done? Like, it's got to be secret. Well, it took a lot of research, but paper after paper, research, it's out there. They're bragging about it. So you can find it, too. And we're going to share all of our, all these documentation so you can look at it, too. It's actually, they brag about how they made it. Pretty crazy.
Yeah.
Is there another slide or no?
No, I think that's
we
it.
give our rest. But the thing is, everybody, and I think Dr. Ardis says this all the time, a couple things about the conference and why it's so unique. Number one, you're going to get all of these slide presentations that we do.
For sure.
You're going to get all the recordings as well. We don't expect you to be a master at it, or it's going to be a little overwhelming, right? Yeah. That's why we're going to help you build that lifestyle with a 14-day plan afterwards. And then what we're going to do is come back for everybody who's registered on October 5th. We're going to put another Q&A together, even if they try to attack us again. We're going to put another Q&A together for all of you that's included with the cost of the conference, so that we can answer your questions and check in with you on how you're getting, how's it going, your building of your new lifestyle. We want you to get this more than anything else.
Solely to empower you.
And I have to say that we've said this before, that there's no evil that man can create, can do, that our God can't overcome. So I just want to remind everybody. And so this isn't just for now, what we're dealing with now, but it's whatever comes in the future too.
Yep. Everybody give a round of applause for Dr. Jen. All right, we're gonna do some live Q&A here for the remaining minutes we have in the studio. But to remind everybody at home, how many of you ever had a wound or a cut or a scrape, and then your mom poured hydrogen peroxide on there to kill it? What were they killing? Why were they using it? To kill bacteria, hoping you wouldn't get an infection in the wound. Hydrogen peroxide kills bacteria. We all know that it does that.
Yeah,
How many of you knew that every cell in your human body makes hydrogen peroxide?
It does.
Every single cell does. Do you know what the body makes when a virus touches the cell? It releases hydrogen peroxide to kill it. When bacteria touches it, the body designed by God is releasing hydrogen peroxide to kill it. Do you know what happens when venom touches a cell in the human body? God designed it to release hydrogen peroxide to break apart the protein structure of venoms. And it is today what they use in all factories around the world to detoxify venoms. They use exclusively hydrogen peroxide. And it's something that the body makes on its own. All right.
So cool. So cool.
All right, so we're going to get to some Q&A here. We're going to make sure we do that. Is everybody enjoying this? Did you get anything that you haven't heard anywhere else? Seriously?
Probably.
I hope so, because we've been working on stuff that we're surprised. They were like, what?
I heard that yesterday,
my
Dr.
God,
Ardis. That's old news, Dr. Ardis, what you just said.
That's because Ely's the genius up here.
Yeah.
All right.
John, are you ready to hit us? If you have questions online, please go ahead and start putting them in the chat or on to Jono's group. Just go ahead and start firing them off. We'll answer as many as we can in the next, 25 minutes?
Awesome. Let me know when you get here.
When they kick us out, we'll give 20 minutes.
20 minutes. 6 minutes.
Oh, did y'all see
7 minutes?
Wait, did y'all see Patriot Street Fighter walk in here?
Did you want to go
Scott
over?
McKay walk in?
Yeah. Did you want to go over time
shoot.
or not?
Patriot
Your
Street
choice
Fighter.
for the questions.
Did you mean acidic?
1, 2,
I said
1, 2, 1, 2.
And then acidic. Yeah.
Are you taking notes? Hold on. See, no.
1, 2, 1, 2.
Thank you for taking
1, 2,
notes.
1, 2, yeah?
That's exactly what
Pretty
we
much
want.
everything that destroys you is positively charged.
10? Okay.
Cation.
Hey, Jono, he's saying 10. We got 10 minutes.
10 minutes.
All right. So If you have 10 minutes, I would say probably one question, then one person answer, next question, one person answer.
Let's just
Your
give
choice.
a 30
Or if you want to all
answer
answer one
How about
question.
that? 30 seconds.
All right, I'll bet you nobody's going to do
Don't
that.
ask Ely a question.
Yeah, right.
Hey, yeah. Everyone is consigned to short answers. I'm sorry, guys. Sorry to be that guy.
SMS text message.
All right.
Here we go. All right, Jonathan,
Cut the
go.
mic.
Go.
Awesome, you guys rock. All right. So, first question. This is Snowy. Question for Dr. Ardis. How do we address diabetes type 2?
Wait, hold on. We're talking about bioweapons. You want to talk about that one?
We'll
That
talk about
bioweapon?
tomorrow, right?
That's fine. I can curate a little
Oh, no,
more.
this is great. Okay, so the
As
question
it
is, how do
relates
we
to
address
bioweapons? Let me curate that. Go ahead.
how do we address diabetes was the question, right?
Sure.
All right, this is
Type
very
2.
important because the World Health Organization just published two. Is that for me? Oh, my wife's
There's
leaving.
things
Okay, she's throwing
flying.
phones at
She's
me.
had enough of you.
Yeah, she's like, we're enough of this crap. All right,
And,
so
and the context here, sorry, an extension here I see in another entry. I had type 2 diabetes, somewhat controlled with diet ketosis with readings American 110 to 150. Then had COVID for the second time. My husband was full-blown diabetic, but he recovered, not me. I cannot get my blood glucose under control. What has happened? How do I need to treat it?
I can tell you right now, inside of your pancreas is what are called beta cells. Beta cells release insulin. Guess what every single beta cell in the pancreas is actually lined with to control turning it on, to release insulin, and to shut it off? They're called alpha-7 nicotine receptors. And bungarotoxin and cobra toxin, found to be the spike proteins of COVID, if they make it to your pancreas instead of getting in your liver, kidneys, heart, nerve system, you name it, or in your brain, if they land on your beta cells in your pancreas, they're called antagonistic to those nicotine receptors. That means it shuts off the nicotine receptor, and it thus shuts off that cell's function. And this goes for every symptom related to COVID.
Yep.
The cells inside your tongue that taste, they're nerves. They have nicotine receptors on them. And when the venom hits it, it shuts off its function. But the venom is bound to it and can stay in your body for 10 years. They publish it. Venoms can stay in the human body after a snake bite, even treating the victim with
I
antivenom.
know,
It will
it's like
stay
gonna
lingering in their body for up to
It's
10 years, creating diseases and outcomes just like diabetes. In fact, during the pandemic, while we were all locked down, I got 2 seconds left in this, 30 seconds. All right, during the pandemic, they were injecting king cobra venom and conotoxins into animals and creating type 1 and type 2 diabetics within 72 hours. And then they reversed it with nicotine infusions. But they didn't tell you that. You're going to learn that at the Healing for the Ages conference. We're going to teach you about that. You are not producing insulin because they have your nicotine receptors shut off on your pancreas cells that make insulin. And we're going to teach you how to get it off.
Yep.
And hydrogen peroxide is one of those things. We're going to teach you.
Yep.
Sorry, Snowy.
Un gars m'a une cotine. Je l'avais vu sur quelque part.
All right, next one. Okay. Giannis, would these bioweapons they're putting out there cause sudden extreme eye conditions / vision problems?
Absolutely.
We have an expert in the audience on
There's
that
actually
subject.
an expert optometrist in the audience who can see all this, but she's not up here. Okay, so
Yes,
Group.
yes, and yes,
Yes.
and yes.
I
for
think
every-
there's 4 yeses,
for-
absolutely.
For every symptom that anyone is suffering from, it's nothing more than an accumulation of poisons, whether it's bioweapon poisons or food poisons or parasite poisons. And by the way, worms, what do they look like? They look like snakes. And if you read the literature on all the worms, they actually secrete venomous, in the literature, it says that worm spit and worm excrement, they literally call it in the literature venomous excretions. So we're being poisoned by multiple different ways. And so if you look at the root cause of any symptom, it's all an accumulation of multifaceted approaches of poisoning the mind, poisoning the body, and poisoning the soul.
But thankfully, there are all kinds
Yeah.
of solutions that we can
there
employ.
are solutions to every single one of
Yes.
these questions,
Solutions
and
to
I
all
will
of
tell
them.
you.
Yeah.
If you go to the NIH's website right now or type it in the Google, type in long-term effects of snake bite envenomation. The NIH published a paper. One of the things after a snake bite injecting venom into a human is diabetic retinopathy that leads to blindness
Yep.
in diabetics. That is a lingering long-term side effect that can show up 2 years after the snake bite, with the venoms just circulating in your body, and it'll just release the retina, and you'll be blind eventually. So yes, absolutely it does it.
I was thinking of something else.
Amazing. People are very interested on hydrogen peroxide. So Dr. Wendy Roscoe asks, how do you know how much H2O2 is good before it is too much oxidation that damages healthy cells? And a continuation is, does peroxide also kill the good bacteria? And then faith and purity, this is all connected. What is the dosage for overcoming long COVID with the food grade hydrogen peroxide?
Dr. Ely.
That is, Wendy was up. That is one of the folks on my research team right there. So she may not want to hear it from me, but my personal stance on it is we know what neutralizes it. And I think it's important if you're going to use hydrogen peroxide that you do a couple things. Number one, it's got to be diluted. Yeah. Number two, it has to be food grade. And we have a guy on stage right here, Dr. Ed Group, whose whole team put together a hydrogen peroxide formula that we can trust. It's at 6%.
Food
all right,
grade.
food grade. And then you have to have specific dosing on it. Now, what you got to do after that is make sure you neutralize it. The two things we know that neutralize hydrogen peroxide are glutathione and catalase. Catalase is an enzyme. So when we recommend hydrogen peroxide, we always say, hey, take it and give it a 2-3 hour work window. And then just to be safe, neutralize it afterwards before you do any more, right? Because hydrogen peroxide actually has a 10-hour half-life. So it can stay pretty bioactive and then can become a problem, an unintended problem as well. But really what it comes down to is if we aren't turning on the natural inherent systems of our body making hydrogen peroxide, we're missing the mark. And that's where the bees and bee pollen and glucose oxidase come in.
Well, they asked
But
if
it is miraculous in cases where people are struggling.
Every
With
single
vaccine
person,
injuries.
yeah, every
long
single
hauler COVID
person
symptoms.
says
So they
that it helps to
asked a
abate
really
it.
good question. Does it hurt the helpful bacteria? But in all the papers and all the research that I found, it only attacks the gram-positive, and that's like E. coli, staph, salmonella,
Enterobacter.
right, Pseudomonas. Like all of those are the ones that it attacks. So the answer is your healthy bacteria is safe.
Okay, amazing. So JP, when on toxin binders, is it safe to drink your own urine?
That's
Okay,
a Jana
so-
Schmidt question.
That's Ed.
I know this one kind of needs a
only
little
Ed.
explanation, but I guess people want to know. Page 144 is the name here, but Dr. Group, is it safe to continue doing urotherapy during an intense parasite cleanse and or full body cleanse, or should you stop urotherapy while cleansing? They're connected, that's why they're
How long do I have to
You have
answer
30
that?
seconds.
Urine it's not a waste product like we're led to believe, and the way that talking about conjugation, like Dr. Ely was talking about, by the time that things come out in your urine, it's specifically highly charged. For example, I just pulled up a study where they took a liter of someone's urine and it produced over 8.5 kilowatts of electricity, which ran a small house for a whole day. There's nothing more powerful from an energy standpoint when we're talking about rebuilding our biofield and putting back the energy and the vibrations than your own urine. Now, I've studied this extensively over the last 3 years and talked to many practitioners all over the world. And they said, they've all told me and shared cases with me saying that, yes, you can see these things and identify these in urinalysis, but they're just pieces. They're fragments. And those fragments in your urine are used as relay mechanisms to come back in your body so your body's energy field, because your urine is energy, can understand and read what else needs to be eliminated and how fast it needs to be eliminated from the system. I searched and searched. I still have not been able to find, maybe over the first couple months, but with urine therapy, you have the first 90 days, which is a detox and cleansing. That's why you don't recommend people going in for blood work, because it's just, your blood is all over the place. It's detoxing and everything's coming out. The second 90 days is more of a regenerative repair, and the third 90 days plus is more when you start getting into regeneration aspects. So I would say that there is nothing that I've found, and no doctor that I've talked to, where someone with heavy metals or anybody that's getting rid of anything inside their system where those numbers have increased over time. Actually, just the opposite. The numbers start going down, which confirms all of the ancient research from 5,000 years ago. By the way, this urine therapy, urine has been used for multifaceted approaches. That it's not dangerous and it's not toxic, and it's what your body creates every single day, the most powerful medicine in the world. Now, that's not, I wouldn't say that's a magic bullet approach, because we need to address many, just like they have many vectors, urine does a lot and covers the majority of those vectors that we'll be talking about, but that's not the only thing. But to answer the question, I would say that we personally have had hundreds of thousands of people all over the world that have been doing this for 2 or 3 years and not seen any contraindications, except you do not want to do that if you're on any type of heavy opiates. Hopefully you're not on fentanyl or anything like that, but there have been, I think, 1 or 2 cases. Nobody died, but they were on high levels of pain medications, opiate-based, and they as they were consuming it back into their body, their body, they didn't die, but they had a little bit of a reaction.
All right, Dr. Ely.
All right, folks, we are running out of time here, and we love you and we love your questions. One of the hallmarks of the conference is we're going to do 90-minute presentations, and then at the end of them, we're going to have 30-minute Q&As, 4 of them each day. So if you register for the conference, we'll be able to do it. We will do our best to take questions from our virtual VIPs as well. So all we can say to you is, please, we can't help you if you're not there. So make sure you sign up for the conference. You're going to get so much. And now we get to finally have the conversation that we need to have as a people to save humanity. Thank you all so much. Dr. Ardis, what you got?
God bless you all at home. We want you here. I believe truly that there's individuals on this earth that were actually gifted by God. If you read in the New Testament, you hear that to some he gave spiritual gifts, like the gift of prophecy, the gift of discernment, and to some he gave the gift of healing. I truly believe there's 4 individuals up here that were given that gift to not only discern, research studies, apply that stuff clinically to their patients, and then apply it to their families. There's a reason why the 4 of us will be never intimidated or scared at all for any future pandemic. And we want you to join us in that effort as a group and as a family. I'm Dr. Ardis. I hope you join us. HealingForTheAges.com.
Amen. Thank you, everybody.
Amen. Thank you.
God bless you.